today we have Debanjana with us, and she's going to talk about proteins and rather how we can build better proteins. So Debanjana, actually she hails from Presidency University in Kolkata, and she's an integrated PhD student at TIFR, and she'll be graduating this year. So, all yours.
Thank you, Lars. So, hello everyone, I'm Debanjana, and I'm done my PhD from Presidency University, Kolkata, and I have done my master's from TIFR and my PhD also I'm doing from TIFR. So I'll just step down to share my slides. Okay. Yeah. Okay. So today I'll be talking to you about the work that I've done and a little bit about what fascinates me as a scientist. So I am fascinated about the world of proteins, and I'll talk to you a little about that. So the title of my talk is whether we can build better proteins. So starting off, I'll ask you a small question. So you see over here that these are normal red blood cells. So can you tell me what gives rise to the color of these red blood cells? Yes, hemoglobin, right. So what is hemoglobin? Hemoglobin is basically a protein. And hemoglobin is an important protein because it's responsible for the transport of oxygen and carbon dioxide in our body. Now, hemoglobin, as I said, is an important molecule. However, in certain individuals, there is a case called sickle cell anemia. In this case of sickle cell anemia, what happens is the red blood cells, which in the above image, you can see there are concave-like shape, but however, they get formed into sickle-like shape. Now, why is this and what can cause this? So before that, I'll talk a little about proteins. So proteins are present in all cells of our body, in all spheres of life, and they are responsible for you to see me. They are responsible for you hearing me, and they are responsible for our breathing. So they are important in various spheres of life. Additionally, inside the cells, there are these motor proteins which can help deliver cargo from one part of the cell to the other part of the cell. So proteins can actually do mechanisms like this where they walk inside the cell. So it's a fascinating field of science. Now, what are proteins, or what exactly is the protein molecule? So like buildings are made up of bricks, proteins are also made up of individual amino acids, which are the building blocks of the protein. So these amino acids are like the beads on a necklace, where the necklace is the protein. If we have all these beads or the individual units the same, the protein would be like this, where there would be no variation, and life as we know it would not have existed. So instead, what happens is there are a variety of amino acids which form the protein. Like there are different people, there are 20 different kinds of amino acids. So the next question that I'll ask you is, do you think proteins are linear in nature? No. Okay. Let's hold on to that for a second. So how are the proteins formed? As I said, these are basically amino acids. Now, amino is a group which is a base, and an acid is a group which is basically an acid. Now, you know that there can be acid-base reactions, right? So similarly, amino acids have these acid-base reactions where two amino acids can join with each other to have an acid-base reaction. And there is the loss of water to form these amino acids. In the boxes, you can see these bonds, CONH bonds. These are actually the peptide bonds. These peptide bonds hold the individual amino acids together to form the protein. Now, great. So, as I have talked to you before, if the amino acids are the same, basically, so an amino acid has three components. As I told you before, there is this amino group, an acid group, and a variable side chain. This variable side chain may be the same. Then you would have the same amino acid repeating, and it would basically look like this. However, instead, in actual scenario, amino acids look like this, where there are many R groups And in fact, there are 20 different kinds of side chains or 20 different kinds of amino acids which can make protein. And these different kinds of side chains, they can interact with each other by various kinds of interactions. As you can see over here, they may be hydrophobic interactions. Hydrophobic means hydro means water and phobic means a fear of water. So they are not water loving, that is fat loving. Basically, there can be hydrophobic interactions, ionic interactions, hydrogen bond interactions, disulfide interactions, many such interactions which can form in a protein. As a result, the protein chain is no longer linear, but it can form beautiful structures like in the case of hemoglobin. Now, so you can ask me questions in between if you have any kind of doubt. Okay, so proteins can also come in various shapes and sizes and colors. In this case, you can see that the hemoglobin protein has a red color. There are other proteins like the green fluorescent protein, where you can see the protein has different colors. This green fluorescent protein was originally found from jellyfish, and these jellyfish have these proteins, and people have done variations in these proteins to have a plethora or various colors in the protein. Now, there can also be other colors in the protein, like the protein that I work with in the lab. It is blue in color and its name is azurin. So proteins can come in various colors. Also, proteins can come in various shapes and sizes. So the largest protein in our body is actually present in the muscle. It is a protein called titin. Now remember, I told you these proteins are made up of amino acids. Titin is a protein which is made up of more than 34,000 amino acids. There may be small proteins. You may have heard of insulin, which people take for diabetes. Insulin has about 54 amino acids. And there may be also smaller peptides, which are, see, glutathione is a tripeptide, which is a smaller peptide. Apart from that, you can also have these proteins called transmembrane proteins, where the transmembrane proteins are the proteins which are in blue. The brown and orange that you see over here, yellow and brown, this is the membrane of a cell, which is basically a lipid, fat. These proteins are within the membrane, and which is why they are called transmembrane proteins. Basically, they are transversing the membrane. These are non-water soluble. As you can see, they are immersed in lipids, basically fat. Also, you can have water soluble proteins like human serum albumin, which is present in the blood. So as you see, proteins can be various types, various colors, and various shapes. And they are responsible for various functions in our body, like you seeing me or hearing me or even breathing. So now let's come back to what I said about hemoglobin. So these are normal red blood cells, and this is the normal hemoglobin protein sequence, which is present in all individuals. However, what happens in the sickle cell anemia condition is that a single amino acid change occurs, which we call as mutation. Now, what causes as a result of mutation is this diseased hemoglobin. And this diseased hemoglobin sequence causes this sickle cell anemia disease. Now, can you tell me at this point in time, what do you think? Are changes in proteins bad? Okay, somebody says no, somebody says yes. Okay, so let's just hold on to that thought for a moment. So from here, we can see that a mutation in a protein can cause a fatal disease, which may be debilitating for the person who has it. Okay, now you might have heard about the central dogma of biology, which is basically DNA or the deoxyribonucleic acid, which is what makes us, that is our genetic material, which tells us how we will look, how we will, like how our phenotype, what we are, that is basically the DNA or the genetic material. From the DNA, a process called transcription occurs, which gives rise to the RNA, ribonucleic acid, and then a process called translation occurs, which gives rise to the protein. So basically, what the protein is going to be made is decided in our DNA. Whether someone has black hair or someone has blonde hair, that is all in the DNA. and that is manifested in the form of the protein which is produced. So, as I had told you before, a person having a correct gene, that is a correct DNA, will suppose a person having A having the correct gene, and there is a person B having an incorrect gene of the hemoglobin. A person A will have the correct protein formed and thus the correct RBCs or red blood cells. However, the person B having the incorrect gene will have an incorrect protein and the hemoglobin will cause the red blood cells to become sickle shaped. So you understand that everything is in the DNA. Now, how can we correct this problem? So two scientists, Emmanuel Charpentier and Jennifer Doudna, they have won the Nobel Prize in 2020 for discovering the enzymes or the proteins which are responsible for genome editing. What does this mean? They have been able to make a protein which is able to correct a gene. Okay, so here, this gene correcting protein, which is called CRISPR-Cas9, it is the hero of the story. But Jennifer Doudna and her group thought, why should we have a hero when instead we can have a superhero? So, as you know that there are older versions of cars, right? But we don't ride these versions of cars anymore. We have newer versions of cars, which are better. So in case of proteins also, they thought that why not make a modified version of a protein? And they have done so by a process which is known as circular permutation. So what does this term circular permutation mean? Now to understand this, here I have put a small puzzle in front of you. So you see that there are three proteins, protein 1, protein 2, and protein 3. Now, can you tell me the difference in between these three proteins? Look closely and tell me what differences can you see? Yeah, please. Okay, so he is saying that there are protein 2, there are two ball-like shapes. Okay. Anybody else? Yeah, please. Okay, color of the balls in all three are different. Okay. Okay, let me tell you here that the colors that I have put here are only for depiction. Okay. So if you look at the shape of the proteins, not at the colors, can you tell me differences? Now let's look at what he said. He said there are two balls over here. Okay. So in the other proteins, are there balls? Yeah, right. Yeah. Yeah. Okay. Now correlate what I told you earlier. A protein is a linear sequence of amino acids, right? Right. Suppose you have a chain of a protein whose ends are here. Okay. The ends of the protein are indicated by the ball. So now can you tell me, suppose the protein is like this and the ends of the protein have the balls, are indicated by the ball. Yeah. So the balls are present at different positions, which means the ends of the protein are present at different positions. Is everybody okay with that? Yeah, right. Okay. So the ends of the protein are at different positions. But if you look at the structures of the proteins, do they seem similar? They seem quite similar, right? Okay. So this is exactly what circular permutation is. And to understand it better, I'll require 6 volunteers. So if 6 of you can come up, yeah, come to the stage. Okay. Slideshow. Yeah. Yeah.
We need 2 or 1.
Okay, great. So now we have 6 volunteers over here, and I'll ask the volunteers at the end, can you wear this on your left hand? Just to hold it. And you can wear this on your right hand. Okay. Remember the balls that I had shown previously? So imagine the gloves are the balls now. Okay. So these two people will be the ends of the protein, and I'll request the rest of you to hold your hands. So if you remember, hold your hands. If you remember from the slide, can you tell me what these bonds are? What is the name of the bond? Peptide bond. Right, exactly. So these are the peptide bonds, and these two ends are the ends of the protein. Now I'll ask you guys to form a circle. Can you form a small circle? Okay. Just stand here. Yeah. So now you see that they have formed a circle. That's why I call it circular permutation. Circular means they are arranged in a circle, and permutation means an rearrange. Okay. Now I'll ask you guys to hold your hands. So what am I doing? I'm joining the termini of the protein, or I'm joining the ends of the protein. Now what I'll do is I'll break this bond. That is, I'm making new NNC termini. I'm making new ends of the protein. So this is what you see. However, you can see that the arrangement is the same, right? How the structure of this protein being formed with individual amino acids being the units, you can see the structure is being same. But if you now hold your hands and form a circle with your face to the audience, can you just hold your hands, keep on holding your hands. And you can, so this is the new termini. So I'll break this bond now. So you just face the audience once. Okay, great. Now, so now what you can see is the original NNC termini are now linked. However, the protein is now, the sequence of the protein is new, right? And there are new N and C termini, or the new ends of the protein. So from this demonstration, you can see why it is called circular, because you have arranged the protein in a circle, and permutation because you are rearranging parts of the protein. Okay? Okay? Thank you, guys. You can keep it here.
Yeah.
Okay. Okay. Now you can see here that I have done the same thing. In the protein A, one, you can see that the individual units are like this. I have just taken the unit A and put it at the last of E. So this is a rearrangement in the protein. So it is called circular permutation. Okay, so we can visualize circular permutation by this also, where you can see this is the original protein. You can put a linker between the N and C termini to add these two and then cleave the protein elsewhere. So you have a new protein with a rearranged protein sequence, and this is called the circular permutant protein. How do we make these proteins in the lab? As I told you, whenever you want to make a protein, you have to look at the DNA. So we make the DNA in such a way that the DNA is now rearranged to give a rearranged circular permutant protein. From here also you can see that this is the native protein, this is the original protein, where the termini are there here, and it produces a protein structure like this. In this case, you can see the permuted protein, where the N and C termini are at different positions, and the resultant protein has a similar structure, but it has a linker in a different region. Okay, so now, yeah, please. Exactly. That's a great question. So, okay, so for the audience, so the question was that the DNA, it determines the amino acids in a protein, but how is the structure of the protein determined? So I'll just go back to this slide. So now I told you that there are many types, 20 types of amino acids, right? And if you look closely at these amino acids, let's see, for example, lysine. Lysine has a positively charged group, and this arginine also has a positively charged group. Okay, let's look at aspartic acid and glutamic acid. So these, if you look at their structure at pH 7.4, you'll see that they are negatively charged. So this aspartic acid and glutamic acid are negatively charged. Now, also note that there are some other amino acids like phenylalanine or tyrosine, which are hydrophobic. As I said, hydrophobic means it does not like water. So these residues don't like water. Now, if you put a protein which is comprising of all these different types of amino acids into water, what will happen is some residues will form bonds like ionic bonds. So you see a positively charged group interacts with a negatively charged group. It is forming an ionic bond. Hydrophobic interactions. So here you see that these two groups don't like water. These form an interaction with each other, which is called a hydrophobic interaction. Additionally, there are some amino acids which contain a sulfur atom, like methionine or cysteine. Let's just go back and see. So you see, where is it? So you see cysteine, it has an SH group. Now this can form a disulfide bond. Also, there may be hydrogen bonds involved. So all these type of interactions, they govern how a protein will fold. Okay. So actually, this is the protein folding problem, that if you are given a linear sequence of amino acids, can you say whether the protein will fold and form a structure? This is called the protein folding problem, which people have been trying to solve for like decades. And it's very interesting and like very intriguing how a protein can fold into beautiful structures like hemoglobin, for example. So here what you would expect is, so on the outside, you would have water, right? So the amino acids which are outside, that is exposed to water, are polar. And the residues or amino acids which are inside the protein are hydrophobic, so that they don't have to interact with water. Okay. 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 So now, suppose you are present at a position A of a hill and you have to reach position B, and I give you a huge ball to take from A to B. How will you reach position B from position A? And I give you a red colored ball which you have to transport from A to B. How will you go from A to B? Can you just let the ball go and then think, yeah, it'll reach? No, right? What do you have to do? You have to apply energy. So suppose you are sitting downhill and you want to transfer something uphill. Then you have to provide energy to let it go to that position. In case of reaction kinetics, also we can say that from a position A, if we have a reactant A and we want to take it to a reactant B, then you have to apply some energy to the system. In the case of proteins, what is the case? You have a folded protein, which is more stable than the unfolded protein. What is the axis here? The axis here on the y-axis is free energy or energy. The x-axis is reaction coordinate. So here for a protein, we have to apply energy to the protein so that it can go to the unfolded state. Okay. Now tell me that what do you think? In the unfolded protein, will the different interactions that I told you, ionic interactions, hydrophobic interactions, will they be still there in the unfolded protein? No, right? So in the unfolded protein, all these interactions break and the protein has a higher energy. Why? Because the hydrophobic groups are now interacting with water. They don't like to interact with water. So the unfolded protein has a higher energy than the folded. So our job now is to take the folded protein to the unfolded protein. Why do we want to do this? Is that suppose we have two proteins here, the original wild type protein and the circular permutant protein. Now, how do you know whether the circular permutant protein is better than the wild type protein? You can try to unfold these two proteins. And suppose one takes a higher amount of heat to unfold, which that you can say that that protein is more stable than the original one. Okay. So what we do, the experiments that we do are that we unfold the protein. How? By inputting energy. How can we input energy into the system? Can you guess? Sorry? Boil or heat the protein. Okay, we can heat the protein. Any other way by which we can take a protein from the folded to the unfolded state? pH. We can alter the pH. Yeah. We can provide ATP. Okay, that may be done, but it might not break the particular interactions which are required. You can also provide energy in the form of heat or in the form of force. So what we do in the lab is that we can unfold or denature. So it's called denaturing the protein, because nature means a native state. So we are denaturing the protein. How can we denature the protein? We can do so with the help of heat. We can do so by adding chemicals, by adding chemical energy, or we can apply force. So in our lab, we apply force to unfold the protein. Now what I'll do is I'll give you two types of ribbons. So I'll give, so you can take one. So hold on to these. I'll so give one each. Yeah. Okay. So now I have given everyone two kinds of ribbons. One is golden in color and the other is of a different color. Okay. Now, can you tell me which of these two ribbons are stronger than the other? Golden one, why do you think so? Because it's cloth, okay. Because the breadth, this one has a higher breadth. Okay, somebody says it's a cloth. Okay. It is compact, okay. You were saying something? Okay, it can withstand more tension. Okay, so looks like a majority of you are in favor of this one, right? So can you do an experiment to show me that this one is stronger than this one? A simple experiment. You can do whatever you want to the thing. Okay. Yeah, you can try to break it if you want. Okay. So what I'll do here is what many of you are thinking to do, but you're probably scared that I'll ask the ribbons back. So you can pull onto the protein onto the ribbons like this. Okay. So now you see one has torn, whereas the other one is retained, right? So what I am doing, I am applying which of these three heat, chemicals, or force? Force, right? So this is exactly what we do in the laboratory, is that we take proteins and we are able to pull them. How? We are able to do so with the help of atomic force microscope. So this is a macromolecular version which I have demonstrated for you. So you see that I am taking this ruler or cantilever as it is called, and I am pulling the protein. I'll show you again. So the tip goes like this, and what we are able to do is, this is a protein molecule. Okay, we are able to capture and hold a protein molecule and pull. Now think about the protein that I had told you previously. There are various interactions in the protein, right? Like ionic interactions, hydrophobic interactions. So these different kinds of interactions break when I apply force to the protein like this. Okay, and by this we can tell which protein is more stable than the other one. So this is what we do in the laboratory. On this I've shown on a macromolecular scale. And so we can tell by pulling the protein that how stable the protein is. For example, if we have the original protein, which is also known as the wild type protein, and we have the circular permutant protein, which is a mutated protein, so we can find energy landscape like this. So it is called an energy landscape. Landscape because it looks like a hill, and energy landscape because we are looking at the energies of the protein. So from here, we can tell which protein is more stable and what changes can we do to make the protein more stable. So with that, we have seen that proteins are present in the body in various places and they perform various functions in the body. Proteins, however, can cause diseases like the case of sickle cell anemia. However, we can correct the protein with the help of gene correcting proteins. And Jennifer Doudna and her group, they have made circular permutant proteins to make more efficient versions of the gene editing protein. And from here, what do you think? Are changes in proteins always bad then? Circular permutation can cause a better protein, right? And can we build better proteins? Yeah. Thank you. So that's what I had to show you for today. If you have any kinds of questions, you can ask me. Yeah. 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 That is right. That is correct. Yeah. But in sickle cell anemia, the person has a lot of problems because these type of cells, they go and accumulate in the blood vessels and cause pain to the person. Resistance to malaria is an added benefit that they get, but it also causes discomfort and pain to the person. So by CRISPR-Cas9, what I have told you, a gene correcting protein, it is possible to cure genetic diseases like beta thalassemia or sickle cell anemia. Yeah. Okay. Yeah. So now suppose, so the question is that, yeah. So the question is that can we design a protein in such a way that we can know what kind of effect it will cause to the protein. So one thing is that if you introduce a circular permutation, which is basically a cut in the protein in, suppose this helix. Okay, so these are called helices and these strand-like things are called beta sheets. If you make a protein and make a change in this helix, the protein structure may break down, which you don't want, right? So you have to do a change in these loop regions. These are called disordered regions or random coil regions. So it is preferable if you do in these regions. One other thing is, suppose hemoglobin, for example. The main center that acts is the heme center, which contains an iron. Okay. If you do any change near the heme center, which is called the active site of the protein, then it will be problematic because the active site is the one which is responsible for its function. If you do anything which might disrupt that function, that will be bad for the protein. Yeah. Yeah. So it has to be designed in such a way that it does not cause any problem to the protein. You can also do a randomly at any position. But then what we do, if you randomly do, there may be some versions of the protein which are less stable, which are not that helpful. Then you just remove those and take the ones that are better for your particular purpose. Yeah, please. Okay, so they call it a circular permutant Cas9. So I'll just yeah, so they called it the circular permutant Cas9. And how do they know it is causing a better function? So Cas9 has been used for various functions. One is it is able to edit a gene. It is able to cut a gene. And by cutting a gene, you are basically knocking off the gene or removing the gene. So that they have screened in different kinds of cells, in kinds of mice, and they have seen that, yeah, this is better, functioning better. So you have to do an assay by which you are looking like, suppose, for example, you have an organism which makes green fluorescence. CRISPR-Cas9, if you make that, it is going to cut the green fluorescent protein gene. And so you make a circular permutant protein. If you wanted to do that function, if the organism does not have green fluorescence, then you know it's acting. Then you know it's doing its function. So like that, you have to do assays to see how it can. It's a trial. Yeah. You can plan it also, but it is called in Frances Alnor, I think in 2021 or so, she got a Nobel Prize. So she got it for directed evolution. So what they used to do is they used to make many versions of a protein, and they used to see can it be better. So they have done so by randomly just mutating and picking the one which is the best. So it's like survival of the fittest protein, kind of like that. Yeah, please. Yes. So in insulin, what happens is the body produces, in diabetes, what happens that there are two types of diabetes. In one, the body produces less of insulin, but the body does require insulin for breaking down sugars. So insulin is provided externally. So when you go to the clinic, they give you insulin, which is basically a protein externally, so that it will be like they're supplementing you with a particular protein that you're lacking. Even in sickle cell anemia, blood transfusion is done, which means that you're applying hemoglobin, which is the protein which the person is probably not able to produce. So that is like benefit of proteins, that you can introduce proteins to the body if they are deficit in nature. Yeah. Yes, it is currently being used, and it is in, I think, the phase 3 clinical trial. So so there is one issue, which is the ethical issue, whether we should do genetic editing on an individual. But because it causes a disease, we are like hopefully going to approve it. It has been in the phase 3 clinical trials, and it is showing promising and surprisingly good results. So I'm not sure about India, but I have seen a study which is done outside. They have done. So the main issues are the ethical issues, whether people will want, like if I tell you that, yeah, gene editing is possible, then you can make an edit to any gene of your body, right? So that is against natural selection. But if it is for a disease and it is curing a disease, I think it is going to be approved by the board, which is concerned. Yeah. Yeah, I'm editing the gene. So yeah, because anything which you want to change in a protein has to be done at the genetic level. Because protein, if you want to make a protein, you have to actually synthesize the protein, which is difficult. But if you make a DNA in such a manner that it is the correct DNA or the required DNA, it's easier, much easier. Okay, so we express in a bacteria called Escherichia coli, E. coli. So actually, E. coli is also present in our gut, but we use a different variant which we use in our lab. No. So then what you do is you inject the DNA or even the mRNA. So you may have heard of mRNA vaccines, right? So mRNA vaccines are basically you add mRNA, and that mRNA would produce the protein. So you can either give the mRNA to the person or you can give the DNA by some manner to the person. Okay. So yeah. 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 That's an interesting question. So so the question is that when we eat pulses or any kind of protein in our food, does that mean we have these chains of amino acids in our food? So the answer is yes. Any kind of protein that you eat, like milk contains casein, then eggs contain protein, pulses contain protein. So all these do contain protein. What happens when we eat them? When we eat the protein, what happens is they go to our stomach, and there are certain enzymes in the stomach which digest the protein. So it breaks the protein into their individual amino acids. So it breaks the protein into these amino acids or smaller versions of the amino acids. So any protein that you eat is a big chain of amino acids. It goes into your body. There are certain enzymes called pepsinogen, which is able to cut a pepsin, which is able to cut these proteins and make them into their individual amino acids. So now you have these building blocks in your body. And when your body wants to produce, for example, hemoglobin, it assembles these building blocks in a certain sequence so that it can make the protein required. So that is what happens when we eat protein. But in the case of insulin, as you had said, we don't want the protein to be individually chopped off, right? We want the insulin intact. That is why insulin is injected into the body and not eaten orally. Yeah. Okay. So amino acids are stored in the cells in the cytoplasm. So, and what they are done is these amino acids are actually hooked onto another unit, which will go to the RNA, and the RNA will then synthesize these proteins. So that is where they are held. How? Okay. Synthetic proteins. Yeah. Okay. So the question is, how are synthetic proteins made in like industry? There are two methods by which you can make a protein. One is chemical synthesis. Chemical synthesis involves individually joining amino acids one after the other. That is easy for smaller proteins, like say 50 amino acids or 20 amino acids. But suppose you want to make titin, which is like 34,000 amino acids. That will take a long time, and that will take a lot of resources. So what we do instead is that we involve E. coli, which is a bacteria. What we do is we just give the E. coli this any particular DNA. Where is the DNA? Yeah, we give the E. coli the DNA so that the E. coli knows that it has to make this particular protein. Okay, so we don't want to make the protein, like suppose we have a 500 amino acid protein. We don't want to synthesize chemically. So what we do is we put the DNA in the bacteria, and the bacteria will then produce the protein of interest. So that is how we usually do in our lab. Okay. So what actually people do? So so the question is that if you want to, yeah. So the question is that what the changes that we are doing in the DNA, can we have a virus that can make the protein and then infect the subject, which is the cell? So this is actually one utilization of vaccines, where what they do is they take a virus like an adenovirus. They take out the genetic material of the virus, what is not required, because that will be harmful for us, right? If that's a virus. They take out the genetic material. They put whatever is required, genetic material, for example, green fluorescent protein, GFP. They put a new DNA. They take the virus. Now, virus has a tendency to infect a host cell, right? And they let the virus infect the host cell. As a result, the virus now has the genetic material of the new protein, right? So it will infect the cell, and the cell will produce the protein. So this is what is done in vaccines, for example. And even if you want, suppose we want to infect E. coli, or somebody wants to infect a mammalian cell or a mice, a small mice that we use for our experiments, we can have a virus, adenovirus, with our required gene of interest, and then we infect the individual with the virus. So that is what is. Okay. Are there? Yeah, you can ask anything. Are there any online questions? Okay. 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 So when we make pulses, okay, that's an interesting question. Does cooking destroy the protein? So what cooking does to the protein is, now remember that the bonds that are in the protein are peptide bonds. Those are covalent bonds. Now a covalent bond is very strong, right? However, the interactions in the protein, like the hydrogen bonding, ionic bonds, these are comparatively less strong as compared to covalent bonds. So when we cook the dal or egg, for example, it unfolds the protein. So the protein does not chop off into the individual fragments, it just unfolds the protein. Only the bonds are broken. Now, what happens when you cook, for example, an egg? The egg changes, right? The white color of the egg, the albumin of the egg, it changes in color. Why does that happen? Because on unfolding, the protein becomes very much prone to aggregation. Aggregation means clumping together. So many of these protein molecules clump together and give you, like if you make curd, for example, you put some lime into curd. What are you doing? You are basically changing the pH of the solution. In a milk, if you're adding lime, so that also changes the pH and it unfolds the protein. As a result, the protein aggregates or coagulates or just comes together, and then you get what you call chana, or chena, right? So that is what happens. The protein unfolds and then it aggregates, but you don't get individual amino acids. That only happens when an enzyme acts, like for example, when you ingest it, when it goes to your stomach, because the stomach contains many enzymes which chop off protein. But that does not happen when you cook the protein. Yeah. Mm-hmm. Okay. Okay. That may be due to any impurities, I guess. Okay. So the question is that when we when we wash pulses, there is a lot of frothing. Why does that occur? So that may be due to any impurities in the pulse. I'm not exactly sure about it, but yeah. 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 Any protein, if that is right. Yeah. Okay. Okay. I'm not exactly sure why frothing occurs in protein. Frothing means there has to be some kind of bubble. Yeah. How does SDS break? Okay, SDS doesn't break the protein. SDS actually unfolds the protein. Yeah. Yeah. Okay, so SDS is basically sodium dodecyl sulfate. You may have heard of this. This is basically like a soap. It is an amphiphilic molecule which contains a hydrophobic group and a hydrophilic group, and is basically like a soap, what soap does. So when this soap detergent kind of molecule comes to a protein, it is able to unfold the protein, and because it has a negative charge, it is able to impart a charge to the protein. So that is what he's saying, it is able to impart a charge to the protein. Yeah. Yeah. It just okay. 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 Yeah. Thanks. That was something I didn't know. Yeah. So that is so he is talking about site directed mutagen. Yeah. So what site-directed mutagenesis is? So it is site-directed mutagenesis. So at a particular site, you can introduce a mutation. So this is what we do, this is what we do in our lab. If we have a protein, we want to introduce one particular amino acid change. We do site-directed mutagenesis to cause this change in the protein. So yeah. It may. So in the case, there are different kinds of proteins. If you take egg, for example, and boil the egg, it does not come back to its original state, right? So that change is non-reversible. But for example, so in like 1960s, Anfinsen and their co-workers, what they did is an experiment. They took a protein called bovine ribonuclease, and they saw that if they unfold the protein and bring it back to the original conditions, it does fold back. So it actually depends on the protein, depends on the sequence of the protein, whether the protein is able to refold or not. Yeah. Yes, definitely. Yeah, definitely. So let's take the example of hemoglobin. Hemoglobin has a heme center which contains an iron. And that iron is responsible for oxygen to carbon dioxide, oxygen transport, carbon dioxide transport. Suppose you remove the hemoglobin, then the protein will be no longer functioning. So that is called the active site of the enzyme. So if the active site is in somehow changed or the protein is unfolded, it will no longer function. So that may happen when enzymes are used reversibly again and again and again. So that is how the protein may unfold or degrade. Okay. Okay. The essential attributes of a strong protein are that it should have many interactions. Like suppose we have a class, okay, and the class has weak interactions in between the students, then the class may break apart very easily, right? But however, if you have a class in which the bonding is very good between the students, then it will be a very stronger class, right? Or like the class would last longer. Similarly, in a protein, if the different types of interactions are stronger and the bonds are stronger, then the protein is stronger. If the bonds are weak, then the protein is also weak. So you can okay. So that's a good question. It may be difficult to digest. So in fact, so I told you it's very difficult to chop off a protein, right? So if you ingest the protein, it may chop off. But what happens to the proteins which are present in the cell? They may need to be recycled. What happens to them? So there is a particular machinery called the protein degrading machinery, which is responsible for taking proteins and chopping them into their individual amino acids. Now, there is a tag called ubiquitin tag. So we work with ubiquitin proteins also in the lab. So ubiquitin is a tag which can, suppose this is a protein of interest which we want to degrade, and this is a ubiquitin tag. Ubiquitin attaches to the protein and takes the protein to the proteolytic machinery where the protein will be degraded. Now, this ubiquitin protein has to be stronger than the protein which is responsible, or otherwise this ubiquitin will also get degraded, right? So in this case, it has actually been seen that the ubiquitin protein is more stable than the other protein, which needs to be recycled in the cell. So yeah, their nature has designed proteins in such a way that it is more strong or less strong, depending on their function. So it all depends on the function of the protein. So okay. So if you want to have a protein which will be tagging and taking the other protein somewhere else, it has to be, first of all, as I said, it has to be stronger so that it does not change itself. If it changes, then what's the point, right? Yeah. And you said GFP, right? So what is GFP? Okay. So you see GFP has a beta. So these are called beta barrels. So it is a beta barrel-like structure. And additionally, very interestingly, so hemoglobin contains this iron center, which is responsible for its red color. GFP, or the unit which is fluorescing, the unit which gives color to GFP, is in fact 3 amino acids. These 3 amino acids covalently bind together to form a new entity, and that entity is responsible for these beautiful colors in the protein. How can we make, so this is green fluorescence, right? But now suppose we want red fluorescence. So people have done mutations to these 3 amino acids or amino acids nearby that these 3 amino acids to have changes that give rise to different colors of the protein. So GFP is actually used as a tagging protein. Suppose you have protein X in the body, but all proteins do not have a color, right? All proteins may not have a color, but you want that protein to have a color. What people do is that they genetically or in the DNA, they have this protein A and they add the gene of GFP. As a result, the protein A will have the GFP as a tag and it will show color. Wherever the protein GFP is, there the protein A will also be. So this is how people also use GFP as a tagging protein because it is so fluorescent and it is, you can see the fluorescence even in suppose jellyfish or other model organisms. You may have seen fluorescing green mice or fluorescing animals, right? This is all done by making GFP encoded in the gene of the organism. Yeah. How gene therapy works? Okay. So what I think gene therapy works that suppose somebody has cancer. Okay. So they are given medicine. Medicines are basically a chemical compound which is given to the person, right? Now, gene therapy looks at targeting the person's gene. Sickle cell anemia, if it is cured by CRISPR, that will also be called a gene therapy because you are targeting the person's gene. Why is my hair black? Because I have a particular protein which causes my hair to be black. Somebody else's hair may be blonde because they have a protein which makes their hair blonde. If you can alter the gene, if you can alter my hair gene in such a way that it becomes blonde, it will become blonde. So that is what gene therapy is. You are targeting the gene of the person as compared to, for example, any other conventional therapies where we give a medicine, which is basically a chemical compound. Chemical compounds don't do anything to your gene. But in gene therapy, that is what is done. The gene of the individual is changed or rectified in such a manner. Yeah, because if you want to make a protein, you have to start at the genetic level, at the gene level, at the DNA level. So, no, fine. It's made in the cell. Yeah, it's made in the body. So for sickle cell anemia, what is done is if you are able to introduce this CRISPR-Cas9 and it is able to correct the hemoglobin of the person, then it can be introduced in the body. So that can be. Yes, that will cause genetic changes. Yeah. Yes. Like, for example, blood transfusion, that is very temporary, right? That will only be like if you have given 1000 red blood cells, that is 1000 red blood cells. They may degrade over time. So the person has to have blood transfusion every now and then, every month, for example. But gene therapy, if you are doing at the genetic level, then it will be long lasting. It will be lifetime. That's the advantage of this technique. Okay. So one thing is that CRISPR is like multifaceted. CRISPR can be used to mutate a gene. It can be used to knock off a gene. It can be used to knock up a gene. So CRISPR itself is multifaceted, due to which these two scientists, they had got the Nobel Prize because it has, and is even like nowadays it's getting like many different applications of CRISPR. Okay. So the cost. Yeah. The cost would come. First of all, it has to be approved by the government that, yeah, you can genetically change, because it goes against natural selection, right? So the government has to say that, yes, it can be used. So there are moral like problems associated. But otherwise, if you want to incorporate, then, for example, in CRISPR-Cas9, you would have to have the CRISPR-Cas9 unit as such. Then you would have to have uh, so you would have to have a facility where, like, suppose Ullas is different from me, right? So then it should be a method by which Ullas's cells are taken out, first of all, then the gene editing is done, and then it is introduced back into the body. So every step of this has an associated cost. And if you want to scale up this, if you want to have 100 people, then also the cost will be there. So all of these have associated costs. Yeah. Yes. Yeah. But if the method is known, then it can be applied to many. Can a protein be imagined without the? Okay. Can a protein be imagined without the nitrogen molecule? Okay. So the nitrogen molecule basically is part of the amino group. And you know, the amino group is the one that makes the peptide bond, right? So that is, I'll say, a crucial unit of the whole protein molecule. So, yeah. Okay. Technically, yeah, that should be possible. Like, for example, vaccines are also there, right? So what mRNA vaccines do, they make the protein required. So if I want to dye my hair black, then I should say green, for example. Then I can inject a protein which makes a green protein, and then it should be able to. Technically, it should be, but then I would have to have every single cell with the. So that's a bit expensive, I think, rather than just dyeing hair. Thank you. Yeah.
Okay, so 2 weeks from now, we'll take you on a virtual visit to the growth lab of TIFR. So we'll be online, and on the first Sunday of August, it's about understanding community and what are the steps of this data. So these are our next upcoming sessions. And on this note, thank you very much.
Thank you.